everyone. Good to
Hello.
see you. Go ahead and type your name in for me when you get a sec. Good to see everybody. Okay. Still waiting for a couple people, but we'll go ahead and get started. Gosh, we're almost to the end of the semester. Hard to imagine, but we're almost there. You are working on a couple of lectures this week, and then next week, and then we're done pretty much, right? I wanted to let you know that I've made a pretty big decision about the final exam in here. So I'm going to go ahead and pull up our schedule for us to take a look at to get started today. All right. So you should be seeing the modules in our Canvas course. You can see here the week of the 19th, which is where we're at. There's slides for the epidemiology material and also for pathogens and how they spread, and of course the lecture quizzes. So looking ahead to next week, if you click on the list for next week, you'll see that we have our next lecture exam, which is lecture exam 5. This exam covers the lecture material since the last exam, so it's going to cover the introduction to the immune system and the vaccination material, the epidemiology, and the pathogen spread, which is, again, what you're working on this week. So it's going to look just like the other exams that you've taken in here, same length overall, same types of questions and so on. Nothing different about this lecture exam 5. But here's the deal. If you look at our schedule, let me go back here. You know that if you look at the schedule, you know that there's a final exam that's scheduled for the course the following week. So next week is lecture exam 5. And then on the syllabus and schedule, it shows us that there's a final lecture exam scheduled for the following week. I've decided to make that final exam optional. So what does that mean? That means that if you have a grade that you're pleased with, you can take lecture exam 5 next week and you're done. If your grade is not what you want it to be, and you would like to have an opportunity to earn some extra points, some more points on another assignment, you can take the lecture exam 5 next week, and then the following week during finals, you can take a final exam. Now, what the final exam will be is a collection of topics that maybe some students had trouble with through the semester, maybe based on questions from all of the lecture exams that more than a couple people missed. I'm going to pull together questions from all of the previous 5 lecture exams. They may not be written exactly as they were on the lecture exams, but they'll be the same basic concept in the question. And I'm going to put together a final exam. So you can take that final exam as an opportunity to, bring your grade up. You don't have to take the final exam, though. You do have to take the lecture exam 5, which is next week. But if you're happy with where your grade stands at that point, you can be done. You don't have to take the final exam. Okay. I'm hoping that what that will do is take the heat off of some of you regarding other courses that you're taking. There are many of you who are doing, I think, very well in the course, and there's no reason that you should have to sit for anything cumulative since you've already done well through the whole semester. Now, the other thing I'm going to do for students who decide that they want to take that optional final exam is I'm going to offer them a chance to meet next week over Zoom and talk through some of the topics that will be on the final. What I'm going to do is put a list together of the topics that might show up on that optional final exam. And we'll spend that very last Zoom meeting talking about that. So you'll be as prepared as you can be for the final. Okay. You don't have to decide right away if you want to take the final exam. You can wait till next week, see how you do on lecture 5. That's fine too. I will expect at some point though, after lecture exam 5, I'll send out an announcement and I'll ask you to let me know if you're going to take the optional final exam. So a week from today in our Zoom meeting, you only need to attend if you're going to take the optional final exam. You only need to come to that meeting if you're going to take the optional final exam. And what we'll do is we will look at and talk about the specific topics throughout the course that you're going to see on the final, on that optional final. I want to get you as prepared as possible for that final. Okay. Does that make sense? Does anybody have any questions about it?
Yeah, I missed first few minutes of the Zoom because I couldn't get out.
Okay, so basically what I announced is that next week, of course, is the last week of classes. And next week you have lecture exam 5, which is going to cover the subjects, the lecture topics since the last exam. So immunology, vaccination, epidemiology, and pathogens and how they spread. That's just going to be, you know, another lecture exam. It's not going to be different from any of the other lecture exams that you've had. You'll notice on the schedule that during finals week, there is a final exam scheduled for the class, but I'm making it optional. So if you have a grade in class that you are pleased with, you don't have to take the final exam. You do have to take the exam next week. You do have to take lecture exam 5, but you don't have to take the exam the following week, which is called the final exam. Students who are really interested in trying to, you know, have one more chance to earn a higher grade, so maybe their course grade will go up a little bit, they can take that optional final. Now, a week from today is our last scheduled Zoom meeting. So next Wednesday, we have a scheduled Zoom meeting, and on our syllabus and schedule, you know, on the schedule that's underneath the syllabus, you'll see that for next week's Zoom meeting, it says review for final exam. So you only need to come to that meeting next week if you plan on taking the optional final exam. And what we'll do during that meeting for the students who decide that they want to take the optional final exam, what we'll do is we'll talk about which topics throughout the whole semester, which lecture topics are going to be on that optional final exam. so you can prepare for that as best as possible. Okay, but you know, if you may already know, that you just don't need that optional final exam because you know you already have a good grade, you're pleased with your grade. You may want to wait and take lecture exam 5 next week and see how you do, and then make your decision. That's fine too.
Do we have lab on Monday?
No, only Wednesday next week. Only Wednesday. And it's going to be about the optional final. So for some of you, this may end up being our last, your last Zoom meeting. Okay. So just give it some thought. Give it some thought about whether or not you think you might like to have that opportunity to take that optional final.
If we take the optional final and it brings our grade down, will you still count it?
What I will not allow it to bring your grade down.
Okay.
Right. I want it to be an opportunity for you to bring your grade up. So if for some reason you decided to take it and it just didn't go well, I won't count it. I won't allow it to bring your grade down. Okay. So give it some thought. Finish up this week's material, which is epidemiology, I believe, and pathogens and how they spread. Don't forget to get those lecture quizzes done. Get ready, get your lecture notes ready for lecture exam 5 next week, and go from there. See what you think. If you'd like an opportunity again to take that optional final. Okay. Remember too that at this time in the semester, this is true for all your courses, not just this one. Take a minute and open up your grades on Canvas. Go through all your grades. Make sure that the grades make sense to you. That I didn't accidentally enter an incorrect grade or something, or I didn't leave something ungraded that you think you handed in. It's always a really good idea to double check at this time in the semester just to make sure everything's in order for you in terms of what you've handed in and what your grades look like. Make sure it all looks complete. It makes sense to you. Nothing looks strange or out of place. Now is a good time to do that, because if in a class, let's say you handed something in and you go back into your grades and you realize you never got a grade for it. That's something you would need to reach out to your instructor and make sure you got a grade for the work you did. So this is a good time to do that a couple weeks before the final exam. All right. So what we're going to be doing today is we're going to be talking a little bit about the coronavirus. We're going to talk about the vaccines, just sort of as a little wrap up for the semester. I would encourage you to take some notes today while we're talking, because there's a good chance that the bonus question on next week's lecture exam, which is lecture exam 5, there's a good chance that the bonus question will ask something about the coronavirus vaccines. Okay. So it's not my intention today for us to become, molecular biology experts and know these vaccines in and out and, 6 ways to Tuesday. But I do want us to know and talk about the basic technology behind the different vaccines, because of course we are using multiple vaccines right now. One of the vaccines has been put on a pause because of some rare but severe side effects. So it's a good idea for us to understand exactly what is in the vaccine and how that vaccine is working. Now that you've had
Quick question.
Yeah.
So I just had it and it was awful. Do you know if there's like a time limit you have to wait before you get the vaccine, or is it safe to get the vaccine now?
So there are, for every vaccine, there are not just the, in other words, not just the coronavirus vaccines, but for every vaccine, the manufacturer will determine through studies when you need a booster and if you need a booster. And that timing is important. So in other words, I can tell you, for example, for the Pfizer vaccine, the recommendation is to get one shot, wait 21 days, and then get a booster shot. So 2 shots, 21 days apart. You can go past 21 days. We don't know exactly how far you can go. We have studied it a little bit, and we know that, for example, if you were to wait 6 weeks between the 2 shots, so that would be 42 days, that's still fine. So anywhere from 3 to 6 weeks, in other words, between the
So my
2 shots.
question is, well, I just had the coronavirus, what, 2.5 weeks ago? Do I have to wait to get my first vaccine?
Coronavirus. Okay, I'm sorry. I thought you said you had a vaccine. It's going to vary. What I would recommend is that you speak to your doctor. You are going to want to be vaccinated, even if you've had a natural infection. But it's going to depend how long you're going to want to wait. Is going to depend on the severity of your symptoms, and your physician can help you with that. That's a great.
Cool, thank you.
But yeah, you definitely want to get vaccinated at some point, even if you've had the disease itself. Yeah.
Yeah, I'm not doing that again. It was awful.
Yeah, it's an awful thing. It's an awful disease. And what's so hard is that it varies a lot from person to person. And that's why there's been so much discussion and misinformation circulating, because the experience of COVID has been different for different people. But yeah, if you speak to people who had a bad go of it, who had a bad time with it, they all tell you the same thing, and that is that it's a bad virus. You do not want to get COVID-19. It's not an easy experience to go through. All right. So I'm going to go ahead and pull up some slides. I don't have a lot of slides for us today. We're really just talking today, but I did find a couple of nice graphics for us that might help us understand some of the more complex technology behind these vaccines. Remember, from the vaccination lecture, remember that vaccines are not huge, complex, mysterious things. There's a pretty short list of ingredients that go into vaccines, right? There's the pathogen, either in whole or in part. You're going to put something in there related to the pathogen. If you recall from lecture, sometimes that means in the past we have used, for example, an entire viral particle that we have disabled or made it so it can't infect us anymore. We've also used bits and pieces of viral particles. And of course, these vaccines that we're using now are based on the bits and pieces model. The other thing that you find in vaccines are preservatives, right, to help increase shelf life. Some vaccines have adjuvants in them. And typically we put an adjuvant in if we need to boost the immune response even more. That has not been a problem with the coronavirus vaccines. And of course, you also have to supply, in some instances, something to dilute the vaccine with if the vaccine is powdered or freeze dried. It's a short list of material. And I think sometimes some people in the public believe that vaccines have all of these chemicals in them, and that's why people have bad reactions to vaccines. But remember, from lecture, we said that when we respond to a vaccine, when we have what people think of as side effects from a vaccine, that's our immune system that's doing that. There's nothing in the vaccine itself that is giving you a headache or giving you a fever or giving you a blood clot or giving you anything. What the vaccine does is stimulates your immune system, which is then doing what the immune system does. It's initiating an inflammatory response, and it's triggering the production of antibodies. And that's where all of those effects come from. The reason you get a fever is because your immune system releases chemicals, cytokine chemicals, that bring about a fever. In some ways, I try to remind people friends and family who tell me, oh, I had my vaccine, I feel terrible. I always sort of joke around with them. I say, well, that's actually kind of good, because that means your immune system is working. Your immune system is doing what it's supposed to be doing, which is responding to that vaccine. It's unpleasant, and in some cases, it's very dangerous, as we talked about in lecture. Some people's immune system responds out of bounds to a vaccine. And for those people, vaccines can be dangerous and even deadly. But the good news for us is that is a very rare occurrence. Even with what's happened lately with the Johnson & Johnson vaccine, which is on a pause now. That means we're not giving it to anyone right now, because there have been 6 women who have had blood clots develop within 2 weeks after getting the shot. So what's happening right now is the Centers for Disease Control and the World Health Organization is reviewing those 6 cases, reviewing the medical records of those women, reviewing whether or not they had COVID before they got vaccinated, whether or not they have comorbidities or other diseases going on, whether or not they've ever had blood clots in the past. They're reviewing the medical record because they're trying to figure out if this effect is real, in other words, caused by this vaccine's stimulation of the immune system, or whether or not it's just a fluke. Remember, something in the order of 6.5 million doses of the Johnson & Johnson vaccine have already been given to people, and 6 women have developed blood clots. So that is fewer than 1 in a million doses. That's less than 1 in a million. Remember from lecture, we consider it a rare and severe effect if it happens in 1 out of a million doses.
Can I ask you a question?
Yeah.
I mean, because there are presumably hundreds of thousands of people every day that get blood clots, and they've had the Johnson & Johnson vaccine. So are they saying that these blood clots are traced directly back to the Johnson & Johnson vaccine?
So certainly there are lots and lots of people that get blood clots every day, but there are not lots and lots of people who have recently had the vaccine who have gotten blood clots, because physicians are keeping a very close watch on that. So for example, over the past however many months since we started vaccinating, if you showed up in your doctor's office or in an emergency room and you had symptoms of an embolism somewhere, pulmonary or perhaps a deep vein thrombosis, your physician would have reported that. And you had just been vaccinated recently, your physician would have reported that to the CDC. It seems strange, but there have only been 6 cases. 6 cases that occurred within 2 weeks of the last of the shot of the vaccine.
Dr. P, could I ask a question?
Yeah.
You had said that those 6 women, they were going to look at their medical history and they were going to look and see if they had COVID.
Yeah.
At
They're
this
going to look at
point-
everything. They're going to look at everything in their medical history.
But at this point, if they've been vaccinated, they would only know if they had COVID before if they were tested at the time, right?
That's a great,
Because-
yeah, that's exactly right. So if they, in other words, when I say they're going to look to see if they've had COVID, they're going to look to see if there's a record of them getting tested and
Right.
coming up positive for COVID. Because you're right, there would not be any way to know now,
Right.
because the antibodies that they have in their blood now might be from a natural infection and might be from the vaccine. You don't
Yeah.
know.
And there's no way to tell.
There's no way to tell.
Okay.
That's a great question. Yeah. But essentially what they're trying to do right now, which is a very difficult job with only 6 cases, they're trying to see if statistically they can show whether or not the likelihood that the immune response to the vaccine created the clot, or if there's some other reasonable explanation for why these women got the clots associated with getting that vaccine. It's a, the other thing I tell people, and I mean this sincerely, is that the fact that they've put a pause on that vaccine right now should give all of us a good feeling, because what that means is they're paying very close attention. They're paying very, very close attention to what's happening to people after they get vaccinated. And doctors are reporting these things to the Centers for Disease Control when they occur. And the public health officials in our country and around the world who are administering these vaccines in different countries, they are paying attention and they are not taking any risks. And that's a really good thing. That should make us all feel good. Because that's what needs to happen when a new vaccine is used, particularly across the globe. I mean, think about that. These vaccines have not been introduced just in our country. These vaccines are being introduced around the world. So it's very important that we are gathering data. Now, you don't have to tell me if you've been vaccinated if you don't want to, but if you don't mind, if you've been vaccinated, put the number 2 in the chat if you got contacted after your vaccine over your cell phone, asking how you felt. Anybody have this experience? Did you get a text from the CDC? I can tell you I did.
I did.
I'll share with you that I've been vaccinated, and I got contacted through the Centers for Disease Control, through a data collection program called VSafe. V, the letter V, safe. And essentially what happens is, Suzanne, you can tell me if your experience was different, but I got a text starting the day after I got my vaccine, basically saying, how are you feeling? And giving me questions to answer. Are you feeling, they would show like happy face. Are you feeling happy face? Are you feeling sad face? And then, do you have any symptoms, and which symptoms do you have? They are gathering data from everyone, or at least I'll say from a random sampling of people who are getting vaccinated, in order to try to learn as much as possible about how this vaccine is stimulating our immune systems. Everyone's immune system is different. It depends on how old you are. It depends on whether you have other health problems. It depends on lots of things, how well you are going to respond to a vaccine. But we know from history that some vaccines tend to stimulate the immune system more strongly than other vaccines do. And that's why people feel lousy after they get them, because of the antigenic properties of that particular virus.
I feel like I've been picked as one of those, one of the samples, because I got, for the first week, like it was the next day, and then it was weekly check-ins,
Yep.
but much more extensive than a smiley face.
Oh,
And
yeah.
And now the state of New Hampshire calls me every week.
That's good.
To check
That's
in. Yeah,
good,
well,
right?
I don't mind. Yeah. So yes, I feel great. And yes, I've had my second vaccination.
For my experience, which was again through the CDC, after the first shot, they checked in with me each day for a few days. Then they checked in with me, after a week or so. Then they said, "Have you gotten your second shot yet?" And I said, "No, not yet, but I'm scheduled." After my second shot, I was contacted, and it was interesting because they asked different questions after the second shot, which tells me that they already have some data that shows that their immune response is different after each shot, which makes sense, right? Remember in the immunology lecture that the immune response after a first exposure to a vaccine is, if you draw it on a graph, the first response is going to be, moderate. And then you get a booster and you get a much more significant response to the vaccine. So it makes sense that for many people, at least with these 2-shot vaccines, because some of the vaccines are only 1 shot, for the 2-shot series, people are reporting, "I didn't feel so bad after the first one, but I felt lousy after the second one." That actually makes sense. Your immune system is going to respond much more robustly after the second shot, the booster shot. And that's what we want it to do, because that's going to allow for your antibody production to be boosted way up. You're going to get more antibodies. You're going to get antibodies that bind to the virus more tightly. And you're going to get longer memory, longer immune memory. So those are good things. You feel terrible during the process, right? You feel lousy for a couple of days, but it's actually a good thing. It makes sense from an immunological standpoint. Okay. So I want to pull up these graphics for us, just so we can talk a little bit about how these vaccines differ. So this is a graphic. It actually comes out of a hospital system down in Florida, but I liked it because it's very, very simple. This was a graphic that they were sharing with patients who were coming into their hospital who had questions about these RNA vaccines. So remember, the Pfizer vaccine and the Moderna vaccine are based on this RNA technology. So what they're showing us in the graphic is they're showing the needle here. I love this. Here's the needle, and they've blown up what's inside the vaccine right here. And they're showing us that in the vaccine injection, there is RNA in here, and there's a coating around it. And then they're showing us that that is going to be given to us, that's going to be injected into our body and taken into our cells. That's what this represents. So the RNA is going to come into our cells, this single-stranded material, and then it's going to be used to build viral proteins, which will then, as you can see from the arrow, stimulate this large, awkward-looking thing, which is your immune cells. And they're also reminding people here that the protein that this RNA is coding for is the spike protein on the outside of this virus. So you have to remember that SARS-CoV2 it's a coronavirus. And like all coronaviruses, that it's enveloped. So it has that, that wrapping of membrane around it. It's a single-stranded RNA virus, which means its genetic material is made out of RNA, not DNA. It has spikes coming off the surface. You've all seen that picture 10 million times that they show on the news with the red spikes coming off of the viral particle. There's the spike protein. There's also a couple of other proteins on the surface of the envelope that are also antigenic. Remember, that word just means capable of stimulating the immune system. So what this vaccine, what this technology, this RNA technology is doing is, instead of delivering the viral particle in a vaccine, it's delivering the spike protein, but it's delivering it as a recipe. It's not actually the spike protein, is it? It's the instructions for making the spike protein in the form of RNA. So in the laboratory, what they've done is they've isolated the spike protein off of the surface of the virus. They've isolated it in the laboratory. They then deconstructed it to determine the sequence of the amino acids in that spike protein. And then they built the spike protein in the laboratory, because we're capable of doing that in 2021. We can manufacture proteins in the laboratory. And from the sequence of the amino acids in that protein, they were able to take a step backwards and figure out what the RNA would look like to build that protein. Remember, the central dogma tells us that in living cells anyway, DNA gets used to make RNA through a process called transcription. RNA then gets used to make protein through a process called translation, and proteins give us our traits. So what that vaccine is a piece of RNA that contains the code to build the spike protein. Now, what Moderna and Pfizer did, and the reason their vaccine is worth, I don't know, hundreds of billions of dollars probably in the long run, what they did was they figured out a way to wrap that RNA molecule in a protective coating of lipid. Because as you know now from our lectures about DNA and RNA, RNA is a very fragile molecule. Remember? DNA isn't fragile, but RNA is very fragile. RNA is not intended to last very long in nature. Cells make it through transcription, then they translate it, then it just degrades, it goes away. So anytime you try to do anything with RNA, you have a problem. And the problem is that RNA is very fragile. It breaks down and degrades very easily. So what Pfizer and Moderna had to do was figure out a way to coat the RNA in something that would keep it structurally sound until it got into our cells. And that coating, that lipid material that they've coated the RNA with, that's proprietary. They're not telling us what it's made out of because that's their special recipe. That's what they're going to make their money from, is figuring out how to keep that RNA molecule intact long enough to get it into our cells and begin this immune response. So that vaccine is very cutting edge in terms of using RNA this way. Now, when you tell people that the vaccine is the instructions, basically, to build the spike protein, sometimes people in the public will say, well, why don't they just put the spike protein into the vaccine? Why don't they just put the spike protein in? They can build it in the laboratory. So why not just put the spike protein in the vaccine and skip the middleman, skip the RNA, skip the process where the cells read the RNA and build the protein? Why not just do the protein? And the answer is proteins are big, complicated molecules. As we know, they have hundreds and even thousands of amino acids in them. And so when you put that molecule into a vaccine, when you put a big protein into a vaccine, you're going to get not of one very specific immune response, you're going to generate a very complex immune response to different parts of the protein. And you're going to get a less directed immune response by doing that. Remember, in the old days, it seems like the old days now, we used to put the entire viral particle into the vaccine. We inactivated it first to make sure that it could not be replicated. But we used to put the whole thing in. We put the genetic material in, we put the capsid, remember the capsid around the genetic material with the spikes coming out or any other structures that were on a particular virus. And the reason that, it works, sure it works, but you're not going to get a clean immune response directed against the best antigens. You're going to get a variety of immune responses against this big, relatively gigantic, complex viral particle. So by using the RNA, the vaccine is cleaner in the sense that the type of immune response that's going to be generated is much more directed. It's directed to the particular parts of the spike protein that we know are the most dangerous parts, essentially, the parts that help that virus get into our cells. So the RNA technology is really, it's genius, really. And it's only been recently, obviously, since the coronavirus, that companies have figured out a way to keep that very fragile RNA intact long enough for our cells to generate an immune response. You've probably heard that the Moderna and the Pfizer vaccines have to be kept in a -80 freezer, right? They have to be kept very, very cold. And the reason they have to be kept cold is because of the RNA. RNA is very, very fragile. And you've got to treat it with kid gloves, or it's going to degrade and the vaccine won't be useful. DNA is a much more robust molecule. DNA, as you know, gosh, we can harvest DNA from dead bodies, right? We can harvest DNA from forensic materials that have been out in the woods for years. We can harvest DNA from the teeth of dinosaur fossils. DNA is a very robust molecule. You don't have to baby DNA, but you do have to baby RNA. And they figured out a way to do it, which is pretty impressive. So this next slide, this is a graphic. Again, it's just designed to show people how this vaccine works, essentially, right? So what they're showing over here, this blue line is supposed to represent the membrane of a human cell. So the human cell would be gigantic. It would fill up, more than your whole screen. We're just looking at a little slice of this human cell here. And then they're showing us this little circle here with a squiggly red line in it. This is what's in the vaccine. This is the active particle in the vaccine. It's a piece of RNA surrounded by lipid, encapsulated in lipid. Again, we don't know what that lipid looks like because that's proprietary. That's secret information that only Moderna and Pfizer know right now. Now they're showing us that these particles that are in the vaccine come into our cells. The RNA gets released, okay? Remember, inside of your cell, any piece of RNA is going to be treated the same way. Your cell cannot tell viral RNA from bacterial RNA, from human RNA. It can't, because the molecule looks identical in all things. So this RNA that got delivered in the vaccine is going to get translated. And that's what they're showing us here. This blue globular structure represents a ribosome. Here's the small subunit, here's the large subunit, and they're showing us that the red RNA molecule is being decoded by the ribosome in the process called translation. And the protein is being built from amino acids. That's what ribosomes do. They read each group of 3 bases on the RNA. Remember, those are called codons. tRNA molecules deliver amino acids one by one. Those are the red balls here. And the red balls, the amino acids, get joined together with peptide bonds, building a protein. And they're showing us that this is the spike protein. And what they don't show you on this slide is what happens to the spike protein next. This graphic is much more complicated. Don't worry about it too much. This is one that Moderna actually produced. But I put it in our talk today because they do a nice job in this central graphic showing you what's going to happen to that spike protein. So this represents, again, a human cell, this big sort of pinkish purple thing here. You can see they're showing you a piece of the nucleus in the human cell. This is cytoplasm here. This is the outside of the cell. Here's one of the particles inside the vaccine, piece of RNA with lipid surrounding it. It's going to come into the cell. The RNA is going to be released and treated like messenger RNA. They're showing us the ribosome as a gold circle. And the ribosome is, as it reads the RNA, it's building a protein. And then they're showing us that protein right here. Remember, that spike protein that the human cell just built, that's viral protein. It normally comes from the virus. So what'll happen is 2 very different things. Number 1, the viral protein, the spike protein, is going to get released. It's going to get secreted out of the cell. And number 2, it's going to be embedded into the surface of the cell. We talked just a little bit in the immunology lecture about the idea that every cell in your body belongs to what's called an MHC group. That's the histocompatibility complex group. In other words, your cells have a certain identity to them. They have a label to them. And when immune cells come by, they interact with all of our other cells, and they basically look at that label and they say, does this cell belong here? They look at the MHC and they go, yeah, actually this cell does belong here. I'm going to leave it alone. What happens with the vaccine is pieces of the spike protein, again, that the cell just built, are going to get presented to the outside of the cell on those MHC complexes. So that blue thing that is sitting on the outside of this cell right now, that's viral. And the next time an immune cell comes through the neighborhood and checks in and says, hey, does this cell belong here? And it reads the MHC, it's going to go, oh, hold on, there's a foreign viral protein right here. That's what they're trying to show us over here. They're showing us that the immune cell, which is right here in pink, is reading the MHC. It's understanding that this is not human protein, and it's generating an immune response. And that immune response will ultimately create antibodies. These vaccines are very, very useful to us in terms of moving forward with the way we vaccinate people. And part of the reason for that is that they are quick to make. If you think about what we used to have to do to make a vaccine out of entire viral particles, we had to grow virus in the laboratory. And you can't grow virus the way you grow bacteria, can you? You know, for bacteria, as we've been talking about, all you have to do is put some of the bacterial cells into some culture medium, put the container into the incubator, and let nature take its course, right? The bacteria will divide. It will grow and make more cells. You can't do that with a virus because viruses can't replicate themselves. Viruses are not cells. They're not alive. So when you want to grow up virus in the laboratory, you have to first grow the host cell in culture. You have to grow a culture that has the host cell in it, the human host cell in this case, which is not easy. It's much, much easier to grow bacteria in the laboratory than it is to grow human cells. Once you have the human cell culture growing, though, the next thing you do is you infect it with viral particles and you let nature take its course. Those human cells will replicate the virus and make many more copies of it that can then be harvested. So you can imagine making a vaccine when you're trying to make it from an intact viral particle takes a lot of time. That's where the original estimates came from during the pandemic, when people were saying, oh my gosh, we're not going to have a vaccine for 2 or 3 years. They were thinking about conventional vaccines. What's amazing about the RNA vaccines is that RNA is really easy to make in the lab and fast. As long as we know enough about the virus that we have had an opportunity to examine the part of the virus that's antigenic, the proteins on the surface of the virus, in other words, once we know the order of the amino acids in those proteins, and that does take a little while, but not nearly as long as other technologies. Once we know the sequence of amino acids, we can backtrack. We can figure out what the messenger RNA would look like to make those proteins. And then we can build the messenger RNA. It's not hard.
Excuse me.
Yeah.
Not to interrupt, but there's like a really bad thunderstorm, so I might lose.
Oh, okay. Yeah, it just
internet,
started raining here too. Hopefully
Yeah,
we won't lose each other.
Just wanted to make you aware.
That's okay.
Thank
Thanks
you.
for letting me know.
Yeah, here too.
So they're much quicker to make, RNA vaccines. And you saw that, right? You saw that these vaccines were made in less than a year. And they're much quicker to tweak. And this is going to be important for this coronavirus because it mutates. All viruses mutate. All viruses mutate. But this pandemic virus, because it's so widespread and because so many humans are copying it every day in their bodies, it's had lots of opportunities to mutate. And there are some very dangerous variants that are circulating now. This virus will continue to mutate until the world reaches herd immunity, if it ever does. So what's nice about these RNA vaccines is that they can be modified quickly. The next version of the Moderna vaccine, the next version of the Pfizer vaccine will include RNA from different variants. It will have the instructions to build the spike protein from different variants. So that is a huge step forward for us in terms of the production of vaccines in a shorter amount of time. But as I said earlier, there's drawbacks to this vaccine, right? The fact that you have to keep it at -80. A
Can I ask a
-80.
question before you move on?
Yeah.
It's more of a distribution question.
Yeah.
When the boosters, quote unquote, are made, or the second round of shots,
Yes.
will they be trying to get us the same shot that we had originally?
So if you've been vaccinated with an RNA vaccine, so again, Pfizer or Moderna, if you've been vaccinated already, you've been vaccinated against the original strain of this virus. You've been vaccinated against the original strain. The mutants that have come up since the original strain first appeared on the planet. Remember, there's a UK mutant and there's a Brazil mutant. They go by those names generally. The differences in those mutant viruses are in the spike protein. So what Moderna is doing right now is building new RNA against the new spike proteins. And in the next round of vaccine, which could be towards the end of this year, maybe next year, we don't really know yet. The next round of vaccine will be able to protect us better against these new mutations. So yeah, if you've been vaccinated against the original variant.
Oh, I get that, but my question was more like, is the CDC going to ask that or ensure that the people who had Moderna before get Moderna again, or
Oh, you mean, so if you've had Moderna, are you going to get Moderna the next time?
Indeed.
Yeah, that does not, as it turns out, that's not nearly as important as you might think it is. And the reason it's not as important is because after you've been initially vaccinated against a virus, subsequent vaccines can be a little different. They're going to be fundamentally different anyway, right? Because we're going to be vaccinating you against a different version of the virus. So it won't matter if you get vaccinated with a different commercial formulation. The fact is you will already have partial immunity from the first round of vaccine. So the second round can come from any manufacturer. We do like, I will say this, we do like when you have to get boosters for your first round of vaccines against any virus. We really like it if you get your 2 or sometimes 3 shots from the same manufacturer. Whether or not there's real science behind that, we don't know. But anytime a new vaccine is given, so everybody is getting it for the first time, we like it if you get your series of shots from the same manufacturer. After that, though, as the years go by and you get boosted, after that, you can get it from different manufacturers. It's not going to make a difference. It's a great question. It's an interesting question.
Could I ask one other question?
Yeah.
Instead of waiting to see if people catch COVID again and how bad it is, are they also testing people to see like the level of antibodies? Because I know,
Yes.
like I had chickenpox when I was a kid, and I either had to get the vaccine again or I had to be tested to see if I had the immunity still.
Yes.
And they did that, and they said, "Oh yeah, you still have the immunity from 3rd grade." So I'm like, okay.
Yeah. Well, there's a lot of work being done right now with regards to people after vaccination. So one of the things that we're trying to figure out right now is how long are we going to make a high level of antibodies? This varies from vaccine to vaccine, which is why, for example, you get a whole series of measles vaccines, for example, when you're a baby. Then you get boosted when you're 5. Then you get boosted when you're 15. Then you get boosted sometime after 65, if needed. That all comes from studies of how long the antibodies get produced, how long the antibodies stay at a high protective level. For some viruses, the antibody production does not last. It lasts for a while, always, but it doesn't last very long for some vaccines. For other vaccines, you're done once you've been through your original series. A good example of this is the tetanus vaccine that we all get. That vaccine gives you 10 years worth of antibodies, which is pretty good, but it doesn't protect you forever. You have to get that vaccine boosted every 10 years, or you're not fully protected. Now, the interesting thing about chickenpox, since you brought it up, is that we know that in most people, a natural infection, so in other words, not a vaccine, but actually getting chickenpox, gives you lifetime immunity for most people. Some people, though, don't get lifetime immunity. That's why when they developed a vaccine against chickenpox, they decided to start checking antibody levels in people. But interestingly enough, most people who get a natural infection with chickenpox are protected throughout their life against reinfection with chickenpox. It just depends on the virus. It's very, very interesting. So we don't know yet how long the interval will be before we all need another shot, because we have to watch people. We have to measure their antibodies every week or every month, and we have to watch for when they start coming down. That's when you want to booster people, when their antibodies are starting to come down, but they're not gone yet. What you don't want to happen is waiting too long and allowing the antibodies to get so low that the people become vulnerable again. Let me, just in the interest of time, just so we get to it, let me talk about the other vaccines that are available, the ones that are not RNA-based. I do want to say that there are 2 vaccines that are in wide use right now around the world that I can't talk to you about in any detail because we don't have information about them. And you might guess one of them is from China and one of them is from Russia. So there are 2 coronavirus vaccines that are being used, again, around the world that we just don't have any information about because their governments don't share that information with us. But in terms of the other vaccines, we do know quite a bit about what's in them. So one of the vaccines is this Johnson & Johnson vaccine. And one of the things that interests people the most, I think, is the fact that you only need one. A lot of people were drawn to this vaccine because they thought, oh, great, it's one and done. I don't have to go back for a second shot. Now, again, what would determine whether or not you needed a booster would be how good the antibody production was after the first shot. For many vaccines, the antibody production after the first injection is just not sufficient, which is why we booster it a few weeks later. Some vaccines require 1 booster shot, so 2 injections. Some vaccines require 2 booster shots, so 3 injections overall. It varies. It depends on the virus and how good of an immune reaction it stimulates. So this is a diagram of how the Johnson & Johnson vaccine was developed. This blue circle represents a human cell. And up here, you can see a picture of a viral particle. Now, this is not the SARS virus, is it? This is not the coronavirus. This is a type of virus called an adenovirus. And if you're not familiar, adenoviruses cause infections in humans, including things like the common cold. But they're also they are also capable, different strains are capable of infecting all kinds of different animals. There are lots and lots of different adenoviruses. This particular adenovirus that they're showing us in the drawing is one that we've been working with for decades in the laboratory. What we did with this virus in the laboratory was we learned how to grow it up, again, in a human cell culture, and then remove its genetic material. So basically all we're left with is the shell. This is not an enveloped virus. This is one of those polyhedral viruses we talked about. You can see it does have spike protein, but it doesn't have nearly as much spike protein as the coronavirus does. It's just a different virus. It's an adenovirus. So in the laboratory, we've been using adenoviruses for a long time to deliver genetic material to cells. Primarily, this is done for research purposes. If we want to make genetic changes inside of a cell, we can deliver the new genetic material that we want to add in. We can deliver it by putting it inside an empty adenovirus. So this is one particular type of adenovirus. And again, it's been developed in the laboratory. So this is not a virus that is any danger to us because it no longer has the genes, the genetic material necessary to infect and cause disease in humans. It's been it's been damaged, it's been modified so that it's no longer dangerous. So what Johnson & Johnson did, which is pretty interesting, I think, is they used this adenovirus to deliver genetic material related to the coronavirus into human cells. So similar to the RNA vaccines, but very different in some respects. What they did was they decided instead of delivering RNA in, they decided to deliver DNA. And the reason they decided on the DNA is because of what we talked about earlier. DNA is a much more stable molecule. So let's think for a minute what they had to do to accomplish this. They had to take the RNA out of the coronavirus. In the laboratory, they had to harvest the RNA molecule that the SARS-CoV2 virus has inside of it. They took that RNA out, and then they reverse engineered what the DNA would look like that would code for that RNA. Remember, all DNA, all RNA, it's the same molecule, whether it's found in a virus or a human cell or an animal cell or a plant cell or a bacterial cell. It doesn't matter. It's the same molecule. And all DNA and all RNA on this planet get made the exact same way. DNA is used to make RNA through transcription. RNA is used to make protein through translation. And it's protein that gives us our traits. So they took an RNA and they did a reverse transcription. They used an RNA to build a DNA. Very unusual. Living cells can't do that. Living cells don't have the enzymes necessary to do that. But we learned how to do it in the lab. And you know how we learned how to do it? By studying a virus, by studying the HIV virus. Because the HIV virus has an enzyme in it called reverse transcriptase. The HIV virus has an enzyme that does exactly this. It uses an RNA to build a DNA. So Johnson & Johnson in the laboratory built a DNA that would code for the RNA that you find in the coronavirus. And that RNA, again, is the instructions to build the spike protein.
Do
By
we
you-
know, I'm sorry.
Go ahead. It's very complex, I know.
Do we know of any other viruses that have that reverse transcriptase?
Reverse transcriptase. Yes, we do. I can't name them for you off the top of my head, but that enzyme has shown up in other viruses as well.
But we only knew to look for it after it was discovered in HIV, is that it?
That's right. And it was found, you have to remember, the HIV, the original epidemic for HIV was back in the early 1980s. That's when that enzyme was first purified from viral culture. And very quickly then we started looking for it in other viruses. We also, of course, used it in the lab. We figured out how to build it and we started using it in the lab. So it's routinely used now to, again, build a DNA from an RNA for all kinds of research purposes. And now for the purposes of building a vaccine. So what happens when you get the Johnson & Johnson vaccine? What's getting injected into your arm is an adenovirus that is not pathogenic. It cannot harm you. It doesn't have its own genetic material in it. Instead, it's sort of an empty shell of an adenovirus. And inside is the DNA instructions necessary to produce the RNA that will build the spike protein. That adenovirus shell still has its own spike proteins on it. And that's what gets it into our human host cells. Those spikes that are coming off of that shell of an adenovirus are what's going to attach to receptors on our cell membranes. And once those spikes attach to receptors, just like in nature, the virus will inject its genetic material into the cell, and that's what they're showing us here. So that DNA gets released into the host cell, and the host cell treats it just like any DNA. It's going to first transcribe it to make RNA, and then it's going to translate it to build the spike protein. The spike protein for the coronavirus, not for the adenovirus. Now, like I said a few minutes ago, this vaccine is just more big, more large, more bulky of a molecule than the RNA vaccines are. So the immune response is not as clean, not as directed, not as specific as the RNA is, the RNA vaccine is. Think about it. There are adenoviruses in that vaccine, the shell anyway of the adenovirus. So you're going to generate an immune response to that because it's foreign. You're also going to generate an immune response to the coronavirus spike protein, which is what the vaccine is intended to protect you against. But your immune response is going to be broader to that vaccine than it is to the RNA vaccines because of the actual physical structure of what's in the vaccine. So that's why perhaps the blood clotting problem shows up even though it's very, very rare, there's a possibility that the fact that this vaccine is delivered in the shell of an adenovirus, it may be triggering an immune reaction that then triggers the formation of blood clots. But we don't know yet.
I mean, are they looking at correlation between, I mean, I
You're
just
absolutely
noticed that it was women of childbearing age. So
Yeah.
is there anything hormonal about the
Well,
reaction?
absolutely. The fact that it's women is important. Women are not men. Sometimes in medicine, they like to pretend that women are men, but women are not men. And women of reproductive age, as you just said, have a very different hormonal profile than men do. And so, yes, we have a different immune reaction to things than men do. The fundamentals are the same. We all have an innate immune response and we all have an adaptive immune response. But the little very specific details of that response are different between men and women. And that's why, for example, women are also more prone to autoimmune diseases, especially in childbearing age. So, yes, you better believe they're looking at that. They're going to try very hard to figure out if, number one, if this is statistically looking like an actual vaccine related immune response, or if statistically it looks like it's just a fluke. There are statistical tests that they can run that will give them that information. And if it shows up as being probably related to the vaccine, to the immune response that the vaccine generates, they will then try to figure out exactly what in that adenovirus delivery system is triggering that response, because they will want to fix that vaccine. If it turns out that there's something about the vaccine that is causing an over response by the immune system, they'll want to fix it. So yes, you better believe they're going to study that. Yeah, it's going on right now, I'm sure. So that's Johnson & Johnson. There is one other vaccine. This one is not in wide use yet. It's in use in other countries, but not here in the United States quite yet. This was the vaccine that was developed at Oxford University. This is the Astrazeneca vaccine. You may have heard of this one too. This vaccine had a little bit of trouble early on in its development, way back, it seems like decades ago now, but way back in December of 2020, when this vaccine was in its early clinical trials. There was another very rare event that happened in the subjects who got vaccinated. It's not on this slide. I'll tell you what happened though. A person who was part of the phase 1 clinical trial, so a volunteer who got vaccinated with this AstraZeneca vaccine, developed a very significant paralysis. I believe it was from the waist down. This person very suddenly became paralyzed. Now, again, was it the immune response to the vaccine? Was it something that was going to happen in this person anyway? Was it a stroke? Was it another infection? They didn't know. It was only one person. It was a man, but they immediately halted the trial. So the AstraZeneca vaccine has been slower to be introduced to the market because of that event. So that was during the very early days of the clinical trials for that vaccine. Now, they went through and they looked at that person's medical record and medical history, and they looked at all kinds of things related to that person. And they ultimately decided that it was not an immune reaction, that something else had happened in that man, and it just coincided with vaccination. and they resumed the clinical trials. So it took a while for this vaccine to get all the way through its phase 3 clinical trials. As far as I know, those trials are finished. The vaccine is reported to be safe and effective, but it has not yet been approved everywhere for usage. So that's why you don't hear about it here in our country. It is being used in parts of Europe and in other parts of the world. So let's take a look at this one. This vaccine has a very similar technology behind it, and it's this adenovirus. You can see that they've actually given the adenovirus a name here. It's called the ChAd-OX1 viral vector. So again, what they did was, like a lot of other people have done, is they developed an adenovirus shell, essentially, that is unable to cause disease in us. They took the natural genetic material out of this virus and created the shell that they could then put any DNA molecule in that they wanted to put in. So just like you see with the Johnson & Johnson vaccine, the Astrazeneca vaccine is a DNA vaccine, and the DNA molecule codes for part of the spike protein that comes from SARS-CoV2. Now, Johnson & Johnson and AstraZeneca sound like the same vaccine, don't they? They sound the exact same vaccine. They are the same technology, but I can't tell you whether they're the exact same vaccine because that information is just not public. We don't know exactly what DNA is in each one of those vaccines because that's proprietary. That's private company commercial information that they don't have to disclose to the world. So it could be that the two vaccines are generating an immune response against the exact same piece of the spike protein. We just don't know. But they are using the same technology. They're using this adenovirus technology to deliver a DNA molecule into our cells so that the spike protein, or at least a portion of the spike protein, can get produced to allow an immune reaction to occur. Other than the issue early on in the clinical trials, I have not heard through my channels, I have not heard of any other concerns about the AstraZeneca vaccine. But because of that early problem, some countries have decided they're just not gonna, authorize the use of this vaccine. It's a shame because, like the Johnson & Johnson vaccine, the AstraZeneca vaccine, because it contains DNA, does not have to be kept at -80. The Johnson & Johnson vaccine and the AstraZeneca vaccine, because they contain DNA, they don't have to be handled so carefully. They don't have to be kept at such cold temperatures. So if you think about it, there, in all of the many parts of the world where it's just not possible to find enough facilities that have the kind of freezer units that are necessary for the Moderna and the Pfizer vaccines. These other two vaccines are going to be very important in getting the whole world vaccinated. There are a lot of parts in this world where people live at a different standard of living than we do. And in order to get those people vaccinated, we need the DNA vaccines because you don't have to handle them so carefully. It's my understanding that the AstraZeneca vaccine doesn't even need to be refrigerated. They're going to call for it to be refrigerated, but in a normal refrigerator. So just a plain old food refrigerator. But I think some studies have been done at least a couple that have shown that that vaccine can sit at room temperature for a while and still be perfectly effective. So both of those vaccines are going to be important moving forward in terms of getting everyone vaccinated, not just the people in the United States. I don't know, Debbie, I don't know of any other vaccines that are using the RNA technology. It's very interesting. At the start of the pandemic, when people realized that the only way out of the pandemic was going to be through vaccination, at the very start, 11 vaccines were initiated. So 11 different companies jumped in and started developing a vaccine. Some of them were RNA vaccines, some of them were DNA vaccines, some were other technology, I'm sure. But originally there were 11. And through the first maybe 6 months or so, a bunch of them said, no, ours isn't going to work. And they dropped out. And then it became a race as to which vaccines were going to make it to market first. And because remember, the most money gets made by the companies who produce the vaccine the fastest. So there was a big push at these companies to develop these vaccines as quickly as possible. That made a lot of people very nervous. But as far as I know, the Moderna, the Pfizer, the Johnson & Johnson, and the Astrazeneca are the 4 vaccines that are intended for coming to market right now. That doesn't mean that there won't be companies that jump in now and start making a coronavirus vaccine.
Can I ask a question about the teams of people that help produce this?
Yeah.
Kind of from top to bottom.
Yeah.
So I'm imagining there's, people who, the geneticists and
Molecular biologists.
Molecular biologists. I was wondering if there's molecular biologists
Yeah.
that are working in there. I mean, because I can imagine shifts of a molecular biologist working on replicating this and examining it and things like that.
You better believe there were chemists on these teams. There were
Sure.
probably biochemists on these teams. There were probably, of course, infectious disease experts. There were probably people who are experts in the coronavirus, not the SARS-CoV2 virus specifically, but coronaviruses in general. They would have been the teams of people would have been from very broad categories of science. And that's because they had to do a lot of different things all at once. I'll tell you, to me personally, the most fascinating aspect of the Pfizer vaccine and the Moderna vaccine is that lipid coding. They figured out a way to put some kind of lipid around that RNA that will not only keep it from degrading, but will help it bind to and get into our cells and not be toxic. And I mean, that's fascinating to me. It's some kind of a nanoparticle of lipid that they have coated this RNA with. And that's new. That's brand new. They got away with not using a viral vector like adenovirus because they developed that special coating. That was probably a lot of chemistry, a lot of biochemistry, and a lot of just basic science behind it, not necessarily virology behind it.
It is super cool.
It is. It's very cool. And like I said, I expect, because we're now pretty sure that we're going to need ongoing vaccination against this virus. This is not a virus that's going to go away anytime soon. It's going to come out at times, just like influenza does, and cause, sporadic epidemics from time to time, perhaps. Hopefully, though, we can get it under control so that it is not like just disturbing everyone's life every moment. we're going to need additional vaccination, in other words. And the beauty of the RNA vaccine, the idea of the RNA vaccine, is that you can make changes to the RNA based upon whatever versions of the coronavirus are floating around at any one time. Just like for the flu vaccine, you know, you have to get inoculated against influenza every year because the flu vaccine is the flu virus is constantly mutating. But the flu vaccine requires that we actually grow up those strains every year, and it takes a lot of time and a lot of money. And these new RNA vaccines are much more efficient to make. So I expect it's not the last we will see of them.
I
So
heard
go ahead.
I heard something on NPR about some of the strains and mutations that they're finding in India right now. And they're mostly the problem is, since they don't even have enough hospitals or places to treat people, they don't really have a handle on what's out there, but
You know,
they're trying to figure out if there are some that have multiple mutations in them. They're saying
yeah.
either more contagious and easier to spread, or more resistant, or possibly both.
Both, right? We know,
Yeah.
for example, that the variants that are circulating right now, including the UK variant, that's the B117 variant you've probably read about. That variant has a change to its spike protein that makes it bind to the receptor on our cells much, much, much more strongly. It's more transmissible. And that makes it worse, right? It's just a worse version of this virus. What's happening in India is the nightmare scenario. What some people forget about a pandemic is the reason that we try to get a hold of a pandemic, other than the fact that we want to save lives, sometimes people forget about that, is that we don't want to overwhelm healthcare systems. When you let a virus circulate uncontrolled, what ends up happening is, because everyone is susceptible to it, it's a brand new virus. Because everyone on the planet is susceptible to it, you get so many infections all at once that their healthcare system gets overwhelmed. It's happening in Brazil right now. It's happening in India right now. There have been more than 1.2 million new cases over the past week in India. Remember
just
how many
worry-
people live in India?
Yeah.
Their
I'm just worried that this is going to be like a different way of carving up the world economically that's going to
Yep.
be even worse than it is now.
It's awful, isn't it?
And
It's
yeah,
in our best interest. It's in our best interest to get everyone vaccinated.
yeah.
But because there is money to be made, we're not going to get everyone vaccinated at the same rate. And that is outrageous. I mean, just from a human rights standpoint, that's outrageous. But from even a medical standpoint, it's outrageous because, again, until the world reaches some level of herd immunity, this virus is going to continue to just rage around the world. It's going to show up as waves of infection. So it's in everybody's best interest for us to get vaccines into the hands of every country around the world so that as many people as possible can be vaccinated. Remember, this virus, all viruses, are completely dependent upon their host in order to persist in the environment. Theoretically, if everybody became immune to the coronavirus, it would go away. People don't understand that, though. They don't have an understanding of the concept of herd immunity. And so we have two problems right now, really. We have a lot of resistance to getting vaccinated. A lot of people in our country and in other countries just don't want to get vaccinated. And we have a problem of countries that want vaccine that can't get it because it's expensive and because there's money to be made by selling it. My only hope is that as the more wealthy countries like the United States get more and more and more of their citizens vaccinated, they're going to be willing to send more vaccine to other countries. But we are in a race right now. We are in a flat out race against this virus right now because of places like Brazil, places like India, places where you have lots and lots of people and nobody is immune. This virus loves that. This virus is just having a field day right now, and more mutations are developing. So only time will tell whether the world is going to do the right thing and, in everyone's best interest, get this vaccine delivered to everyone instead of just, trying to turn a profit on it. It's really outrageous.
I know that China and Russia aren't sharing information with us, but I would hope that we're sharing information with them.
That's a great question. The answer is we are, but we're doing it indirectly. In the United States and in countries like the United States that have open democracies, representative democracies, we allow scientists to publish freely. So all of the work that gets done in this country in research laboratories anyway, not in commercial businesses, but in research laboratories, all of that information is shared. The scientists in China and in Russia, they don't have that same freedom. They're not free to share with the world what they're studying in their research labs, but they can certainly get access to ours. So all they have to do is read the journal articles and read what's online and read what is being posted publicly by research scientists. The commercial companies, though, that make these vaccines, so Pfizer, Moderna, Johnson & Johnson, AstraZeneca, they're not going to share what makes their particular vaccine unique. They're not going to share that information with anyone, because that's what makes their vaccine commercially valuable. So you better believe, like, for example, Pfizer is not going to tell anybody what that nanoparticle lipid coating is made out of. They're not going to. I'm sure they've already patented it, and they will have an exclusive patent on it for some number of years. So that, again, we are a profit-driven, capitalistic society that way.
Down with big pharma. Just kidding.
Well, it's hard. It's just hard. There are times.
There's so many things that you need the amount of resources and the money that's behind the research.
I'm sure that, the Moderna and Pfizer will turn and they will say, listen, it cost us billions of dollars to develop the vaccine, and we need to get that money back. Right.
And it's not just the research they're doing now, it's research they did years ago too.
Exactly right.
And that's the part that a lot of people don't get, but
that's exactly right. And thank God for those companies.
I know.
Because they're the ones that are doing the work and making the breakthroughs. Mm-hmm. But unfortunately, it means that in other parts of society where those kinds of companies don't exist, there is less access to the wonderful breakthroughs that we have. Mm-hmm. And what happens in a place like Russia and a place like China is simply that the government clamps down on the scientists and the commercial companies and restricts what kind of information they can release. But what will happen eventually, if it hasn't happened already, is that the United States will get its hands on the Chinese vaccine. It will get its hands on the Russian vaccine, and it will basically reverse engineer it to find out what's in it and see what they did for their vaccinations. The data coming out of China right now is not good. It's showing that their vaccines are not nearly as protective as our vaccines are. So that's not good. That's not good news, because China is another place with many, many, many people in it, and we want them all to be immune. So that data, the early data, is not good. The Russian vaccine appears to be better, at least so far. It appears to be more protective. And I can tell you that Russia is selling that vaccine to lots of countries that are in the Middle East and in parts of Asia. So a lot of people around the world will be getting the Russian vaccine as well. So it's all a big experiment, folks. It's just a big experiment, and we are living in the middle of it. So we all just have to stay tuned. We have to stay tuned to see how well these vaccines are going to do the job, and how quickly we can get to a level of herd immunity, which will push just the circulating amount of virus way down. That's what we need to achieve. We need to give that virus no safe harbor. We want to get to the point where there's no body, no literal human body that it can get into because of immunity. Good questions today. I think vaccination science is very interesting. A lot of people don't realize that vaccines were one of the greatest advancements in medicine. We have achieved the saving of millions and millions, if not billions, of lives through vaccination. So while there's a lot of misinformation about vaccines out in the world, and there are a lot of people who are very frightened by vaccines, we can only hope that, with education and with time, people are more willing to trust the science and get vaccinated. All right. Any other questions?
Maybe you should share your lecture, because you explain it so much better than anyone out there.
Oh, I'm so
'Cause
glad. Thank you for saying that.
Because they're just not answering questions, and just saying, this is what I'm telling you to do, and it's backed by science. But if they actually explained it to people,
I know.
it would be a lot better.
I feel that way too. It's interesting. There's different schools of thought, and my son can tell you that from day to day, I sort of walk around the house yelling into the universe when I hear stories about missed opportunities of chances to teach people something about these vaccines that are just missed. And it's frustrating, because I want to believe, I personally want to believe that some of the hesitancy around getting vaccinated it's not political, it's not ideological, it's just genuine fear. They don't want to get something they don't understand. I understand that, right? People don't want to get injected with something that they don't understand. That's very scary. And I wish that we could do a better job in the public health sector, communicating more, to educate people, like you said.
Because even like the autism, vaccine connection from how many years ago?
Yeah.
And
Yeah.
still people think that,
that's exactly right. It's very frustrating. It's very frustrating. And, the truth is that there is a percentage of the population that isn't interested really in learning. But I want to believe that most people are receptive to learning more and understanding more about things like vaccination. Yeah, that episode around the question of autism was one scientist who did one study that was discredited and pulled from the literature, and look at the result of that. There are still people every day who will tell you, oh, I'm not vaccinating my children. What if they get autism from it?
Right.
Yeah, it's so hard.
Of course, I
really
think
hard.
more people get information from Jenny McCarthy than anyone else.
From Facebook, right? I mean, from TV, from,
Like
I
MTV,
know,
yeah.
I know it's really hard. I will tell you one thing, when I went to get my vaccine, I felt very emotional. I felt very grateful. And I was just like blubbering to the lovely fireman who was giving me my shot about how grateful I was to be able to get, this miracle of modern medicine, this terrifically important part of preventative healthcare. Yeah, he was looking at me like I was crazy.
There were a lot of people like you though, because I got to help give the
Very
vaccines and
emotional.
people were so excited
Yeah,
coming in.
I was like, isn't this wonderful? Isn't this amazing? And he was looking at me like, okay, lady, just roll up your sleeve.
Yeah, I was a total dork about it.
Oh, goodness. All right, everybody. That's all I have for you. I've kept you a little bit late. I apologize. Give some thought about whether or not you might like to take that optional final. You don't have to decide right away. Take a look at your grades, make sure that everything looks in order. And I will be in touch with you next week. Okay? All right.
Thank you.
All right, everybody. Have a good evening.
Good night.
Good night.